Good day, dear students. I'm excited to have you back on our virtual biology lesson brought to you by IOTA Virtual Learning. I remain your biology virtual tutor, Mr. Kehinde Adeshina. In the lesson of today, we will be looking at a very vital process in living things that ensures that living things continually be in existence and not go into extinction. So, get your writing material and come along with me as we dive into this important process. Every living organism is capable of producing young ones which are either the same or somewhat similar to them. Can you guess what this process is called? This process is called reproduction. And in a simple term, reproduction is the process whereby living organisms give birth to young ones which are the same or similar to them. Now, when I say living organisms, it includes humans, animals, plants, and even microorganisms. Reproduction in living things is divided into two types, which are asexual reproduction and sexual reproduction. Let's look at asexual reproduction. Asexual reproduction is a form of reproduction that do not involve gametes. In this reproduction, a part of a parent plant can develop into a new one, or a single living organism can split into two or multiple pieces to develop daughter cells. So, what are the forms of asexual reproduction? Asexual reproduction can exist in the following form: fission, budding, spore formation, or vegetative propagation. We will look at these forms of asexual reproduction as we go along in this lesson. But let's look at what sexual reproduction is. In contrast to the other type, it involves gametes. So, male gametes and the female gamete comes together in a process called fertilization to form a zygote. Now let's look at the first form of asexual reproduction called fission. Fission is a process whereby a single matured cell splits into two or multiple pieces, and each pieces can develop into a new living cell. It will interest you to know that organisms produced from this form of reproduction are genetically identical, therefore they are called clones. Let us look at binary fission. Binary fission is a form of asexual reproduction in which a matured unicellular cell splits into two following the division of the nucleus to produce two genetically identical cells. It is the main form of asexual reproduction in unicellular organisms such as amoeba, paramecium, euglena, and chlamydomonas. Let us look at binary fission in amoeba. For binary fission to begin in amoeba, the amoeba withdraws its pseudopodia and become a spherical shape. The nucleus then splits longitudinally and divides into two daughter nuclei. These daughter nuclei move towards opposite poles. This is followed by elongation of the parent cell. A fission appears at the middle of the cytoplasm between the two daughter nuclei, dividing the cell into two equal halves. So, two daughter amoebae are produced from a single parent. One of the daughter cells have the original contractile vacuole, while the other develop a new one. The entire process of binary fission is completed in less than an hour, although this is determined by temperature. Paramecium also reproduce by asexual means involving binary fission. The first step is the division of the macronucleus into two by mitosis. This is followed by the division of the micronucleus also by mitosis. Mitotic spindle then appears along the micronucleus. This is followed by constriction of the cytoplasm and a subsequent formation of a new contractile vacuole in the two cells. Finally, the paramecium cell divides completely to form two daughter paramecia. Now let's take a look at budding. Budding is a form of asexual reproduction in which a parent cell develops an outgrowth called bud, which later detaches from the parent cell to become a new living cell. This form can be found in yeast and hydra. Let's look at budding in yeast. If you view a solution of warm water, sugar and yeast powder under the microscope, you would observe that in yeast, a little cytoplasm accumulates at one side of the cell and the bud is formed. The nuclei divide by mitosis into two and one of the nuclei enters into the bud. In yeast, the bud forms rapidly and forms a chain of yeast under favorable conditions. Finally, the bud detaches from the parent cell and becomes a new genetically identical yeast cell. This process allows for rapid population growth and its characteristics of yeast species like Saccharomyces cerevisiae. Budding in hydra. Hydra is one of the simplest multicellular freshwater organisms, about 1 centimeter long. It has a tubular body with a basal disc for attachment, a head-bearing mouth, and surrounded by stinging tentacles, a reason it is called many-headed serpent. Hydra reproduces asexually by budding. At the time of asexual reproduction, small bud appears at the body wall. As the bud elongates, a mouth opens at the free anterior end, followed by the development of tentacles at the base of the holder region. The newly developed hydra detaches from the parent body and fixes itself on a substratum to begin a new life. You may pause this video to get the diagram of the process of budding in hydra into your notes. Let's look at another form of asexual reproduction called spore formation. Spore formation is a method of asexual reproduction which is found in non-flowering plants such as fungi and some bacteria. Here, the parent cell produces hundreds of spores from their spore sac in the sporangium, which grows into a new plant. A spore is a tiny spherical unicellular body surrounded by thick walls. It falls to the substrate and grows when conditions are favorable for germination. Bread mold seen on the spoiled bread grows when spores of rhizopus settle on the bread. Let's look at the form of asexual reproduction in higher green plants. In higher green plant, asexual reproduction involves a single parent plant producing numerous identical plants. This is achieved by a process called vegetative propagation. Let us discuss this magic of plants reproducing by vegetative propagation. Vegetative propagation is the production of new individuals from the vegetative part of the parent plant. The vegetative part of plants involved include the stem, roots, and the leaves. This vegetative part can be planted under favorable conditions to form a new plant. This method could be natural or vegetative propagation. Natural vegetative propagation involves the use of natural parts of the plant such as the roots, stem, and the leaves to propagate a new plant. For example, the use of tubers in potatoes. Tubers are underground storage organs which are planted to produce new plants. Tubers are used to propagate a new potato plant. Potato tubers consist of various buds from which new plants can arise when planted. Another example of natural vegetative propagation is the use of bulb in onions. So in onion, bulbs are used to propagate a new onion plant. Bulbs are underground stem of the onion plant, and they are used to propagate a new onion plant. Another examples of natural vegetative propagation is production of runners. Runners are creeping stems from which identical daughter plants can grow. For example, this method is seen in the propagation of strawberry plants. Strawberry plants are being propagated from the runners creeping on the surface of the soil. In ginger, new plants are produced from underground stem called culm, which grows into a new identical ginger plant. In bryophyllum, reproduction is through the leaves. The leaves of bryophyllum contains various buds on the margin. When these buds falls on moist soil, they give rise to a new bryophyllum plant. Now let's look at artificial vegetative propagation. The various forms of artificial vegetative propagation include grafting, cutting, layering, and tissue culture. In grafting, the twig of one plant called the scion is kept over the cut stem of another of the same species called the stalk. Then they are tied together. The tissue of the stalk and the stem combine to form a new plant. This method helps to develop a high-yielding and disease-resistant plant. In cutting, a healthy young branch of a stem of a plant having buds is cut off and placed in a moist soil. The cutting develops roots and grow into a new plant. This is how cassava and sugarcane are being propagated from stem cutting. In layering, a young healthy branch is bent down towards the ground and covered with moist soil. After some time, root develops from the covered part. This is called the layer. This branch can now be cut off from the parent plant to form a new plant. In tissue culture, a piece of tissue is cut off from the growing tip of plant and kept in a nutrient medium under controlled condition. The hormone present in the nutrient medium help in cell division and leads to the development of roots and shoots. These plants are then grown in the moist soil to produce new plants. Using the artificial vegetative propagation method is very useful to farmers. This is because it enables production of new plants in a very fast way and requires little attention during the early stage of the plant development. It also helps to produce high-yielding and disease-resistant plant species. As usual, let's test our knowledge of what we have learned so far by looking at the biology quiz from the past WAEC question. The first question we'll look at is WAEC 1998 question 5. Which of the following can be used for vegetative propagation? A. Ginger stem. B. Yam leaves. C. Plantain roots. And D. Potato tubers. Think about the options and pick your answer. The correct option is A. Ginger stem. Ginger is propagated from underground stem called culm. B is wrong because yams are produced from tubers. C is also wrong because plantain are produced from their suckers. And D is also wrong because potatoes are produced from underground storage organ called tubers. Let's look at question 6 from 1998. Which of the following is a method of artificial vegetative propagation? A. Creeping stem. B. Layering. C. Culm. And D. Rhizome. Think about the options and pick your answer. The correct option is option B, layering. In layering, a young healthy stem is artificially bent down towards the moist soil, where it develops roots and produces a new plant. Option A is wrong because creeping stem is a natural vegetative propagation method. Option C is also wrong because culm is a natural part of a plant and underground storage stem. Rhizome is also wrong because it is a natural part of a plant. Let's look at question 12 from 2015. A plant that undergoes vegetative propagation by means of leaves is A. Hibiscus, B. Bryophyllum, C. Crotalaria, and D. Spirogyra. Think about the options and pick your answer. The correct option is option B, Bryophyllum. Bryophyllum is propagated by the buds on the leaves that falls on the ground to become a new plant. Option A, hibiscus, is wrong because hibiscus is propagated by stem cutting. Option C is also wrong because it is not propagated by leaves. Spirogyra undergoes sexual means of reproduction called conjugation. Now let's look at question 7 from 2020. The plant illustrated above is A. Stem tuber, B. Bulb, C. Stolon, and D. Runner. Look at the diagram critically and pick your answer. The correct option is option B, bulb. The diagram above shows the underground stem of onion called bulb, which is planted to propagate a new onion plant. It is obvious from the diagram that the plant is not a stem tuber, neither is it a stolon nor a runner. All these are different from a bulb. Lastly, let's look at question 6 from 2021. The diagram below is illustration of a fungus. Study it and answer the question that follows. Now the question. The part labeled 1 is associated with A. Sexual reproduction, B. Bud formation, C. Asexual reproduction, and D. Gamete formation. Look at the diagram critically and look at the options to pick your answer. The correct option is option C, asexual reproduction. The part labeled 1 is the sporangium. The sporangium contains spore sac which produces spores in a process of mitosis. These spores are asexual units which falls to the substrate to develop a new plant. All other options, option A, which says sexual reproduction, is wrong because spores are not sexual units. Option B is also wrong because this is rhizopus and it does not produce bud. And option D, gamete formation, is also wrong because spores do not form gametes. That will be all for this lesson. In the next lesson, we will be looking at sexual reproduction in details and also look at meiosis, which brings about the formation of gametes. I hope you have learned something tangible from the lesson of today. Do well to read further on this concept and try answering other questions from your past WAEC to get yourself acquainted with the concept. Till I come your way next time, do not stop reading.